Everyone, welcome to another Global IMMUNOTALK. I'm here with one of the co-organizers and our Global Immune speaker today, Dr. Henrique Veiga-Fernandes. And before I introduce to you, Henrique, I'd like to remind you that next week, Florence will be our Global Immune speakers, another of our co-organizers, so I hope you can join. And so let me stop sharing and now have the delight to introduce to you Dr. Henrique Veiga-Fernandes. Henrique graduated in veterinary medicine at Universidade de Lisboa in Portugal. He was awarded a PhD in molecular and cellular biology from the Université René Descartes in Paris, France. And then he developed his postdoctoral research Institut Necker in Paris, France, and at the National Institute for Medical Research in London. Then came back to Portugal, where he started his own independent laboratory in 2009 at the Instituto Medicina Molecular in Lisboa, Portugal. And in 2015, he became a member of the board of directors at the institute. So Henrique's laboratory focuses on defining lymphocyte sensory mechanisms in health and disease. He uses an integrative, across-level approach to elucidate the tenets of lymphocyte sensing and communication across and beyond the organism. As you know, this is an important area of research because a mounting body of evidence indicates that in addition to their well-established developmentally regulated immune cells are also controlled by dietary signals and neuronal inputs. But however, it is still unclear how lymphoid cells perceive, integrate, and respond to those environmental cues. And this is exactly the area where Henrique's team has made significant advances. And these advances include groundbreaking contributions to the understanding of immunological memory, innate lymphoid cells, and hematopoietic stem cell biology. So
So I think there was a problem with the connection from Alina. So I'll take it. I think from here. So Alina, thank you so much for these kind introduction. I'm pretty sure that you're going to join as soon as possible, as soon as your connection come back. And it is really my big pleasure to share with you data that we've been pushing forward in the lab that really relate to how the nervous system and the immune system interact and communicate with each other to actually, at the end of the day, contribute to a normal regulation of body physiology. I see that Ellen is back to us.
Oh, I'm so sorry. I'm not sure when it got.
It doesn't really matter. So I already thank you for the kind introduction, and we can take it from here.
Sure, that people got to know all the awards you have learned, but I hope
not really, not really.
I just wish to want to say that Henrique has been really distinguished with many awards, and congratulate him for the Sean Zuckerberg award he got last year, among the many ones he received. Anyway, I apologize for the problem with connection, but I think that we still have time, Henrique, to ask you the question, so the audience can get to know you better. Okay, and
sure.
the question for you today is whether you could share one of the most impactful decisions in your scientific career and its implications.
Well, I think that one is actually quite easy, because for me, clearly was the decision to become a scientist. And really that started when I decided to do a PhD. You know, I trained as a veterinary medicine doctor. So my knowledge in terms of science and curiosity-driven science was basically non-existent. And while I had it as an intimate desire, I always remember being sitting in the classrooms and asking, how do they know how the cells communicate? How do they do it? I mean, what kind of experiments do they do? And everything started there. And I think that's important for every single scientist, without a doubt. But when I made that decision, actually, it was not only deciding to do a PhD, but also deciding to do a PhD and go abroad. And the reason for that was that I was actually fascinated by immunology. And I considered at the time that, you know, in my home country, Portugal, I didn't really had laboratories that could offer me the conditions to explore my curiosity. So I would say that while it was the first step into the world of science, was certainly the one that had the most important impact. Because if the decision wasn't that one, I would not be here discussing with you the data we've been generating in the laboratory. So I would say that, yeah, my PhD was the most important decision I ever made in terms of my career progression.
Perfect. Thank you so much, Enrique, and how, yeah, curiosity can be such a driver, right?
Absolutely.
Towards developing a research career. Yeah, thank you so much. Okay, so if you can share your screen, we're going to hear your talk.
I will do that immediately.
Perfect.
Go, and then I'll try
Excellent.
to get here the laser pointer, and I think we're ready to go, right?
Yes.
Good. So thank you very much, all of you that joined today, or if you didn't join today, that probably will then grab the opportunity to watch this talk on YouTube. So what I would like to share with you today is work we've been developing over the past few years. We're now based in Lisbon at the Champalimo Foundation, where we're really pushing forward a certain number of research lines that go around a major or central topic that relates on how the nervous system may regulate immune functions and, most importantly, immune fitness. And this all started actually with an assumption or interrogation that I had from a long, long time. I mean, as immunologists, and I'm not an exception to that, we often look at we have an immunity-centric perspective of immunology. We look at immune cells as effector cells that are ready to go whenever we get an infection. And we often also look at these immune cells isolated from an organ or a tissue context. But actually, the way immune cells develop immune responses or deal with a cancer cell, for instances, is actually far from being a silo, an isolated phenomenon. Immune cells somehow having to interact with very complex environments, with multiple type of cues, as depicted here in this beautiful painting from Hieronymus Bosch. And those cues actually can be very diverse. And they vary not only throughout our lives, they will be different in the fetal life, in the infancy, or in adulthood. But very importantly, those multiple cues will also be quite different from organ to organ. And the way immune cells integrate those cues, I consider one of the most exciting questions likely in the next decades for us scientists and especially immunologists to sort out. Now, one of the families of immune cells that has been discovered, or at least coined more recently, has actually properties that led us to become very excited about them, because essentially they are tissue resident. So they are very abundant in tissues, but not so much in classical lymphoid organs. So this family of cells is what we have coined as innate lymphoid cells. As I said, they are very, very abundant in non-immune tissue. So they are very abundant in peripheral organs such as the intestine or the lung or the adipose tissue or even the kidneys. And similarly to T helper cells, they can be subdivided here in three main families: type 1, type 2, and type 3. And this is due basically to a combination of different transcription factors and cytokines that they can produce. So it comes to no surprise, type 1 ILCs, that includes NK cells and helper ILC1s, can produce vast amounts of interferon gamma, and therefore they're very important in the context of, for instance, viral infections. Type 2 ILCs that produce type 2 cytokines, including IL-5, IL-13, and antiregulin. So it comes without saying they're quite important in the context of allergy or helminth infections, classical type 2 infections. And finally, type 3 innate lymphoid cells that are main producers of innate IL-17 and IL-22. And as you know, these are very important pro-inflammatory cytokines. So it comes to no surprise that they have been involved in inflammatory conditions or also in the immune responses to extracellular bacterial infections. Now, the other important point, in contrast to T cells, they don't have professional antigen receptors like a TCR or a BCR, and therefore they normally get activated by host-derived cytokines and alarmins. If you want to activate, it's just an example, very efficiently an ILC2, you need a cocktail essentially with IL-25 and IL-33 that is normally produced by cells in the tissue, and this would lead to activation of ILC2s that then express large amount of cytokines. And actually, this is very similar to all ILC subsets. So they really, because of where they sit, they are on the front line of immunity. They can respond very fast in a very immediate manner. But there were also some early evidence that these cells, in addition of producing these effector cytokines, they were actually also very important to regulate host physiology. And perhaps one of the most important findings, and early ones, indicating the importance of these cells in general physiology, came from a period of our lives that I call the pre-birth world. And in this pre-birth world, when the vast majority of us are still in the womb of our moms, a lot of things are happening already in our organism. And ILCs are part of that. This is an example of what happens in an embryonic intestine, an embryonic day 15 and a half intestine. What I'm showing you here, this is very old data, but I think very visual. ILC3s that actually colonize the intestine at day 15 and a half, and they are absolutely critical for the development of secondary lymphoid organs. So they contribute to development of, in this case, of the mouse. And why do they do that? Is that in 24 hours from day 15 to day 16, now these cells that were dispersed along the intestine, they coalesce, they get together, and they form the primordia of the structures that will be forming the Peyer's patches in the intestine, important secondary lymphoid organs. Exactly the same thing happens for the development of lymph nodes. So it comes to no surprise, the work from multiple groups has demonstrated that if you genetically modify animals not to have these innate lymphoid cells type 3 in the embryonic period, now you end up with animals without secondary lymphoid organs. So at the time, this was a transgenic mouse that I developed during my postdoc. I was actually very puzzled, but at the same time very excited when I took some of these animals and we started to analyze the behavior of these cells. And these are actually two movies that I'm showing you here in the intestine, day 15 and a half of the embryo. And you can appreciate that these cells actually in the wall of the gut, they send projections from one side to the other, like sensing the environment for local cues that potentially will make them aggregate and form the primordia of Peyer's patches. So I got very excited with these, and my you know, my obsession at the time was to identify potential environmental cues that could lead these fetal innate lymphoid cells to operate and form secondary lymphoid organs. And actually work that we've developed in the lab, and then other laboratories also developed complementary studies, we demonstrated that in the embryo, actually there are maternal-derived micronutrients and also other tissue-derived cues that are actually very important for the function of these cells, indicating that in addition to the classical hematopoietins and alarmins that these cells respond to, they actually can sense very important environmental factors that then condition their function and the development of secondary lymphoid organs. So the central question then became, well, if this is happening in the embryo, then likely in adulthood, the same thing is happening, especially because in adulthood, certain organs become far more complex. Just think about the intestine, you know. Once we are born, we are very rapidly colonized by a complex panoplia of microbiota, both bacterial, viral, and fungi commensals that are very important and actually trigger and control our immune system, but also from the diet. The other aspect as well that is very important is that these immune cells that are, for instance, in a mucosal barrier of the intestine, they will have to interact with a lot of other neighboring cells that will range from epithelial cells, muscle cells, or eventually even vascular cells. So one of our central questions became, how can these immune cells that sit in the intestine or at mucosal barriers can perceive, integrate, and respond to very complex tissue cues? And there we really started a significant number of projects where we started to try and understand how discrete immune cell subsets could integrate signals from other cell types. And one of the cell types that we got very excited with is depicted in this image here, this confocal image. So what you're seeing here is a transversal cut of an adult intestine. You would have one intestinal villi here and another one there. Here will be the crypt, the intestinal crypt. And below the intestinal crypt, we have these beautiful clusters of immune cells. These are clusters of innate lymphoid cells type 3. We call them cryptopatches. And they're very important because they produce IL-17 and IL-22 and these two pro-inflammatory cytokines, in addition of promoting inflammation. They're also very important to regulate epithelial reactivity in the intestine. But what we got very excited was that if you now make a double reporter mouse where you can directly visualize ILC3s and these cells here in red, these are glial cells or neuroglia in the intestine, you can appreciate that actually these neuronal cells send these stellate-shaped projections inside the cryptopatch. And actually, they establish very close relationships with immune cells, suggesting that actually, potentially, we have here an ongoing crosstalk between neuronal cells and immune cells. We found this for ILC3s. This was all published, so I'll not go through this. And we also found this in ILC2s. Actually, if you take a gut, this is also true in the lung. The ILC2s that you find in the wall of the intestine, and they will be depicted here in cyan, 1, 2, 3, 4, 5, 6, and so forth. You've got zoom of some of these areas here and here. You can actually appreciate that all these innate lymphoid cells type 2, so producing essentially IL-5 and IL-13, sit in very close proximity to the local neurons. And all these neurons are cholinergic, because if you now do it with a genetic mouse model, where you drive expression of RFP with this tree-driving line of cholinergic neurons, all of these cells sit in the cholinergic neurons. So I think the central question is, if they are close together, then do they communicate? And if they communicate, what kind of tools, molecular tools, do they use? And I'll give you just a fast example. All this has been published. So we actually found that in these intestinal innate lymphoid cells type 2, they express very high levels of this neuropeptide receptor that is called NMUR1. So a receptor for a neuropeptide called neuromedin. You can appreciate it on this side that this neuropeptide in the intestine and in the lung is similar. It's only expressed by neurons. And very significantly and selectively, NMUR1, the receptor for neuromedin, is only expressed in ILC2s and their progenitors. This is also validated in human, indicating that potentially we would have here in our hands a molecular axis that could be used by neurons to directly communicate with ILC2s. And indeed, they use this to do it. So we tested in a very simple set of experiments. If you purify innate lymphoid cells type 2, and then in vitro, you stimulate them with these neuropeptides for a very brief period of time, you immediately have upregulation of type 2 cytokines, irrespectively of whether your ILC2s come from the gut or of the lung, very rapid upregulation of IL-5 and IL-13. This is true at RNA level. This is even more pronounced if you do it at protein level. And this activation of ILC2s with NMU, with neuromedin, tightly depends on the expression of NMUR1 on the surface of these cells, because if you repeat the experiment with ILC2s from an NMUR1 knockout background, there's no upregulation of IL-5 or IL-13, as you can see here. So neuromedin is a very potent activator of these cells, and the question is, can that be harnessed in vivo? And it can, and actually the phenotypes are rather spectacular. Actually, if you take animals that you now infect with the helminth Nipastrongylus brasiliensis, and you basically create two groups, one of them treated with PBS control and the other one with neuromedin, you can already look at the color of the lung, the top one with a lot of hemorrhage, and you can see the erythrocytes here, while if you now treat the animals with NMU, you preserve essentially the structure of the organ. There's almost no hemorrhage. You can see it by the histology. These activation, induction or injection of NMU leads to very strong type 2, innate type 2 response that downstream recruits eosinophils, mast cells, and as a consequence, there's a dramatic reduction in the infection burden. So all in all, and to summarize a long story and work from our group and other groups, I would say that actually these two stories that I briefly mentioned, they put forward a concept that we baptized as neuroimmune cell units, whereby at discrete areas of tissues and organs, neuronal cells and innate lymphoid cells can interact. That interaction is highly selective and specific. In the case of ILC2s, this operates via NMU, neuromedin, and NMU receptor 1. This leads to activation of ILC2s and elimination of infection. In the case of ILC3s, this relates to activation of glial cells by commensal products that leads to the expression of GFLs that activate ILC3s and in turn expression of IL-22. And these neuroimmune cell units are really poised to ensure mucosal homeostasis and health, and that allows the organism to respond in a coordinated manner with neuroimmune responses that very rapidly establish homeostasis and eliminate the infectious aggression. And a lot of other groups in the meantime have actually shown that these neuroimmune cell units are present not only in the intestine and in the lung, but actually can find them in other tissues, including the skin or the adipose tissue, which really led us to hypothesize some time back that perhaps in addition to these tissue and organ-specific neuroimmune cell units, perhaps there are systemic neuroimmune circuits, in other words, organismic neuroimmune circuits that can contribute not only to fight infection, but potentially also for organismic homeostasis and physiology. And so we started to address some of those questions. One of the first examples of systemic cues that we started to analyze were actually to concentrate on systemic circadian regulation. So we all know that the Earth rotates, and that gives us a period of night and a period of day with light. And a lot of our behaviors are actually controlled by those light-dark cycles. We call that the circadian clock. And what circadian clocks actually allow us to do as mammals is allow us for sensing, anticipating, and adapt to those daily environmental changes. So clearly day and night or light and dark cycles are very important to regulate some of the physiological processes of the body because they are integrated by the brain in a very distinct area that is called the suprachiasmatic nucleus, and I'll come back to that a bit later on. But for instance, in mammals, we now also know that the vast majority of the clocks in peripheral tissues can actually be induced by feeding regimens. And we also know, for instance, and this is critically important for an immunology audience, that the microbiome also conditions body clocks of the host. So the first question we actually asked was whether or not these innate lymphoid cells that sit at mucosal sites could actually follow a circadian pattern of oscillations. And the answer was immediate and quite simple. They do. So I'm giving you an example here of intestinal innate lymphoid cells type 3. On the top row, you've got genes that are responsible for the clock machinery in any cell of the body. ARNTL, clock, periods, and NR1D1 are some examples. You can immediately see that in a diurnal manner, at different times of the day, we call them the ZT hours, they oscillate. So you've got some of them that oscillate with one pattern, the other ones the opposite ones. So they clearly oscillate throughout the day in a diurnal manner. And this is true not only for the classical circadian clock regulators, but also for genes that confer the identity to these cells. And I'm just showing you here a couple of those genes that actually are responsible for the identity of ILC3s. These are essentially transcription factors that are required for ILC3 development and function. You can see that these transcription factors also oscillate throughout the day and the night, indicating that there's potentially, in terms of the biology of these cells, a very important role for the circadian clock. So we actually addressed what that importance would be. And in order to address that, we actually took advantage of this master regulator of the circadian clock, ARNTL, and we specifically deleted this gene in raw gamma T expressing cells. So essentially here in ILC3s, also some subsets of T cells. And what we observed here clearly was that whenever you ablate ARNTL in ILC3s, in other words, if ILC3s don't know the correct time zone within the body, you basically disrupt the homeostasis of these cells in the intestine. They virtually almost disappear from the intestine. You can see it in all ILC3s, across all ILC3 subsets, and also on the ILC3 subsets that express the pro-inflammatory cytokines IL-17 and IL-22. Now, one might ask, well, is this relevant for the life of the animal? Well, it's not only relevant, but it's vital. Because if you now take those animals where you don't have a circadian clock in ILC3s, and you infect them with an attaching-effacing bacterial infection, in this case, this is a Citrobacter rodentium infection, which is the equivalent of the enteric hemorrhagic E. coli infection in humans. You can see that animals without an intact circadian clock in ILC3s cannot control the infection when compared to their litter-mate controls. You can beautifully see it here, the burden of infection of these animals. And as a consequence, these animals die actually very rapidly upon 2 weeks after infection, while their counterparts behave perfectly well and they survive beautifully well. So really indicating that this ILC intrinsic circadian clock machinery can shape the gut defense. But ILC3s do more than just providing a defense against this type of bacterial infection. Actually, they're also responsible for controlling the expression of lipid transporters in epithelial cells in the intestine. And the way this operates, this was beautifully shown by Ron Germain, is that the IL-22 actually inhibit expression of these epithelial lipid transporters. So it comes to no surprise that when you look at the epithelium of these mice, we observe that when, again, ILC3s do not know their exact time zone, now we have increased epithelial lipid transporters in these animals, and this beautifully correlates already in steady state, no high-fat diet, no nothing, normal Shaw. These animals already have increased gonadal fat and subcutaneous fat. Therefore, indicating that an intact circadian clock on ILC3s is absolutely critical to regulate not only intestinal defense, but very importantly also to regulate fat storages within the body. So one of the central points, as I referred, is then what are the environmental signals that actually are providing the exact time zone to ILC3s? So the first hypothesis that we immediately raised was this probably relates to microbiota, because ILCs are in the intestine, just beneath the crypt. It would make a lot of sense. We knew that the microbiota also has circadian oscillations. So we actually tested animals with antibiotics against the control. And then we analyzed the oscillations in ILC3s using this reporter mouse for this very important molecule of the circadian machinery, period 1. And actually what we observed was that while there were some adjustments on the circadian oscillation of ILC3s, they certainly did not invert, not dramatically modified the oscillations of ILC3s. So microbiota was not certainly one of the major cues driving these oscillations. Not only microbiota wasn't doing much, but altering and modifying, inverting feeding regimens was not provoking or leading to an inversion of the oscillation. That's, for instance, what happens when you do this in skeletal muscle or in the liver. Actually, when we did that, we observed a slight difference in terms of amplitude, but certainly the oscillations here are not dramatically modified and not really inverted. But what really fully inverted and immediately inverted the oscillations of ILC3s was when we started playing around with light-dark cycles, that you can see that very rapidly, actually, these ILC3s in the gut immediately invert those oscillations. So we concluded, actually, from these experiments, and obviously, needless to say, we actually did all possible combinations of these three main environmental stimuli. So ILC3s can actually integrate both local, and those would be essentially microbiota and feeding, local cues, and systemic cues, but they will then act in a hierarchical manner. So while microbiota and feeding regimens can adjust some of the parameters of the oscillations of ILC3s, the light-dark cycles are the ones really entraining the oscillations of ILC3s, which raises the hypothesis that likely, since dark cycles are mostly integrated in the brain, in the suprachiasmatic nucleus, that brain signals perhaps directly or indirectly may control intestinal ILC3. So we tested that. And we did it in multiple ways. This has been published, so I will not give you the full approaches. I'm going to just show you stereotactic surgeries that we did in mice in order to specifically ablate the suprachiasmatic nucleus. So this is shown here with MRI image in the coronal view and sagittal view. These were the sham-operated animals. This is the suprachiasmatic nucleus ablation animals, where you can see that the SCN was ablated here and there when compared to the sham controls. And we did these in period 1 venous mice, so we could, in those mice that were surgically operated, then we could look at the ILC3s in the gut and ask whether or not they would maintain an oscillation. And the answer is that whenever you disrupt the SCN in the brain, these cells do not oscillate, indicating that the brain-derived cues can regulate innate lymphoid cells type 3. So as I told you, this was published quite recently, indicating that in the specific context of intestinal ILC3s, light-entrained but also brain-tuned circadian circuits can regulate ILC3s. And this is critical for gut health. In multiple aspects, it regulates how the epithelial reactivity genes control the intestinal microbiome and the oscillations of the microbiome. It controls enteric defense, and I showed you the example of the Citrobacter Citrobacter rodentium infection. And very importantly, I didn't spend much time on these. It also controls locally the lipid metabolism of the host, and mostly because it actually controls how our own organism extracts lipid products from the diet. And so that really, the fact that at a systemic level, these neuroimmune circuits can regulate a lipid metabolism led us to hypothesize that perhaps more general, the metabolism of the host could be regulated as well by neuroimmune interactions. And so we started actually to focus and interrogate the possible existence of neuroimmune circuits that could potentially regulate obesity. And if we're dealing about, with obesity, we likely have to look to two main, I would say, adipose depots. One of them is subcutaneous fat. That's what gives the appearance of these sort of fluffy bacchus. But also, and probably more important so, to visceral fat, that, as you know, is the type of adipose depot that has more dramatic consequences in terms of metabolic health of an individual. So visceral fat is really detrimental for multiple conditions, predisposing for chronic inflammatory pathologies and oncologic disease. And in mice, the vast majority of these visceral adipose tissue is actually the gonadal adipose tissue. So the first question we asked in the gonadal adipose tissue was whether or not this tissue was innervated, and being innervated, what was the vast majority of the nerve fibers that actually would be present in the gonadal adipose tissue. And actually we found, as depicted here, with this marker TH, that actually is a surrogate marker for sympathetic neurons, we found that the gonadal adipose tissue harbors a very dense network of sympathetic neuronal fibers. And interestingly, this gonadal adipose tissue is actually also contains a very significant population of innate lymphoid cells type 2. So the question was obvious. If we harness and we modify the sympathetic tone, in other words, the activity of sympathetic neurons, can we modify the function of adipose ILC2s in the gonadal or visceral fat? And to that hand, we use a lot of different approaches. The first approach I want to share with you is actually a loss of function approach. So we took animals where we performed chemical sympathectomy, what we call chemical sympathetic ablation, using 6-HDA. And what we observed was that upon treatment, ILC2s present in the visceral adipose tissue of mice had a very significant reduction of type 2 cytokine expression, reduced IL-5 and IL-13. We complemented this chemical sympathetic ablation with a genetic ablation of sympathetic neurons. This time, what we've done was to breed actually a Th3 driver, so expressed only in sympathetic neurons, with a ROSA26 diphtherotoxin receptor. So in other words, we're going to drive expression here of the diphtherotoxin receptor exclusively and selectively in sympathetic neurons. And upon administration of the diphtherotoxin, these neurons will die. And as you can see, we basically phenocopy the phenotype we had with 6-HDA treatment. There is a significant reduction of IL-5 and IL-13 expression from ILC2s. Importantly, and quite interesting, we got the exact opposite phenotype if you now use gain of function manipulations of the sympathetic nervous system. So if you activate the sympathetic tone, and we can do it in multiple ways, chemically using clenbuterol, so an agonist of one of the receptors downstream of sympathetic signaling. You can see that once you treat the animals with clenbuterol, there's a fast and strong upregulation of innate IL-5 and IL-13. and also with the genetic approach, this time where it took advantage of chemogenetics. So we drive the expression of DREADs selectively on sympathetic neurons by this breeding. A TH3 with a ROSA26 activates DREAD. And once we activate these neurons with DREADs, now we get an activation of ILC2s with increased IL-5 and IL-13. So clearly indicating that these sympathetic cues can regulate the ILC2s that sit in the visceral adipose tissue of mice. So the central question is, how is this operating? So is this a direct interaction between sympathetic neurons and ILC2s in the visceral adipose tissue? Well, to test that, what we've done was a simple genetic experiment where we deleted the beta-2 receptor on lymphocytes. And then we asked, by the deletion of this receptor on lymphocytes, whether or not we would see an impact on ILC2s. And as you can appreciate from this data, there's absolutely no difference of cytokine expression in the gonadal adipose tissue of these mice, indicating that the effects of sympathetic innervation on ILC2s is likely to be indirect, not directly operating on ILC2s, but perhaps on a third cell type that integrates sympathetic cues and then coordinates and controls the function of ILC2s. So we embarked on an endeavored expedition to identify what those cells could be. And the first thing we did was actually to address the expression of this important beta-2 receptor on different cell types that are present in the visceral adipose tissue. And this is depicted here. In some of those cells, we found actually that there's a subset of mesenchymal stem cells that express PDGFR alpha, that express very high levels of beta-2 when compared to the other cell types. Not only that, but we also found that these cells, these mesenchymal stem cells, were present in very close proximity to neighboring sympathetic neurons, suggesting that perhaps these cell types would be relevant to integrate the sympathetic tone. So we addressed this experimentally, and what we've done was actually by deleting beta-2 specifically on mesenchymal stem cells expressing PDGFR alpha receptor. So we made this breeding PDGFR alpha 3 with beta-2, and bang on, what we found on those animals is that we recovered the phenotype that we observed when we ablated sympathetic neurons, indicating that these neuromesenchyme signals certainly regulate ILC2s. So the sympathetic tone provides a adrenergic receptor signal to mesenchymal stem cells in the visceral adipose tissue that in turn control ILC2s. How could that operate? So what are those signals? So we tried actually to identify what these signals could be. And basically what we've done was to treat animals with 6-OHDA, so no sympathetic input. And then we purified mesenchymal stem cells from the visceral adipose tissue of these animals. And we asked what kind of genes are regulated. So this is just a heat map of genes that were downregulated upon sympathetic ablation. And what we found with that chemical inhibition was that there was one gene that attracted immediately our attention. This gene is called GDNF, and it's a gene that was very highly expressed by these mesenchymal stem cells and was significantly downregulated without sympathetic input. We confirmed that the expression of this gene, GDNF, was also downregulated in a genetic model where we block sympathetic signals in mesenchymal cells by deleting beta-2 and PDGFR alpha, and this is selectively regulated in mesenchymal stem cells. We don't find it in other subsets of mesenchymal cells, nor adipocytes, nor endothelial cells, indicating that the sympathetic cues regulate mesenchymal stem cell-derived GDNF. Now, GDNF actually stands for glial-derived neurotrophic factor, and it's actually a ligand for a very powerful tyrosine kinase receptor called RET. And actually, we investigated and interrogated whether or not RET was expressed in ILC2s in the visceral adipose tissue, and it is, it's expressed. And therefore, we asked whether or not ablation of RET expression in ILC2s could result in a reduction of function of these cells. And indeed it does. So we initially bred this with the VAV1 that deletes basically red in all immune cells. ILC2s from these mice had reduced type 2 cytokines, but then we did a more selective experimental setup where specifically delete red in IL-5 expressing cells, so in this case, in ILC2s, and we made sure that these were in ILC2s by breeding it in addition to RAG1, so T cells are not affected whatsoever. And once again, we recapitulate this phenotype. Loss of red expression in fat ILC2s results in impaired type 2 response. And the opposite type of experiments is also valid and important. If you make a mutant to get a gain of function activity in a rat, where basically more signals go through this molecule, now you end up by having more type 2 innate cytokine production from the rat meant to be gain of function model. So indicating that this neuromesenchyme axis actually is likely regulating ILC2s via these GDNF rat signals. And once again, the question is this relevant for the life of the animals? In other words, what is the impact for the normal physiology and metabolism of those animals? Well, the impact is actually quite important. Not only in what we would call a loss of function model, so what we've done here was actually to take RAG-MSC animals that do not have ILC2s and then transplant ILC2s either from a competent or incompetent or deficient rat ILC2s. So basically in that animal model, we're going to have ILC2s that can respond to GDNF or ILC2s that cannot respond to GDNF. And as you can see here, animals that receive ILC2s that cannot respond to GDNF, now they cannot actually mount, they actually increase in weight very rapidly when exposed to high-fat diet when compared to the controls transplanted with cells of wild-type origin. So not only there's an obesity phenotype, but there's also an important metabolic phenotype because the glucose homeostasis is disrupted, and as expected, the weight of the gonadal adipose tissue or visceral adipose tissue is also significantly increased. Importantly, if you do the opposite experiment, now what you do is to transplant wild-type ILC2s or rat gain-of-function ILC2s, animals with an increased ILC2 function via rat now are protected from developing an overt morbid obesity when compared to their controls. And as you can see, they very rapidly control glucose levels when compared to their controls, and they have a reduced gonadal adipose tissue weight as well. So clearly indicating that the signaling axis, GDNF rat, is very important to control adipose composition and the metabolic homeostasis in these animals. So how is this all operating? So we know already that we have, I would say, a three-part cellular axis whereby sympathetic neurons provide signals to mesenchymal cells that in turn, by the production of GDNF, can activate and regulate the function of ILC2s that, as we know, then activate energy expenditure in adipocytes. So in order to understand how this is integrated, we started by interrogating what was the identity of these neurons that innervate visceral adipose tissue in mice and that apparently could control the activity of ILC2s and the composition of visceral adipose tissue. So we initially started by a monocynated viral approach where we basically injected a virus that cannot jump through synapses, right, in the visceral adipose tissue. And then we asked what kind of neurons could we label with this approach. So the first thing we observed was that actually there was a nerve fiber that actually merges or projects to the gonadal adipose tissue that was immediately labeled. This fiber actually is part of the genitofemoral nerve, And you can see here that all those fibers are essentially sympathetic. And these nerves then project to a very defined ganglion, neuronal ganglia, that is called the aortico-renal ganglia. And you can appreciate here the cell bodies of those neurons, all of them that were labeled with this tracing approach, all of them are sympathetic, indicating that the cell bodies of sympathetic neurons that are present in the aortico-renal ganglia likely control the activity of mesenchymal cells in the adipose tissue. But not only these cells are present in the aortico-renal ganglia, but we were also curious to know whether or not these neural cell types could connect to high-order circuits, so in other words, to the brain. And to do that, what we've done was an approach where we actually, by performing polysynaptic tracing, now we're using virus that can cross multiple synapses, and we injected these virus either in the gonadal adipose tissue or directly in these aortico-renal ganglia, and this is depicted here. And then we compared what areas of the brain would be highlighted. And we always found, actually, that there was a very discrete area in the brain that is called the PVH that was labeled, indicating that cell bodies of neurons that are located in this very important area of the hypothalamus, the PVH, actually connect to the aortico-renal ganglia, that in turn connects to local mesenchymal cells and control ILC2 function. And the reason why we got very excited with these is that the PVH is known to control the vast majority of the sympathetic outputs out of the brain to the periphery. So this really strongly suggested the existence of these long-range circuits starting in the PVH, then passing through the aortico-renal ganglia that then really activates ILC2s in the gonadal adipose tissue. So to formally test that, we actually tried to harness these circuits functionally to demonstrate that we would potentially have an impact or not on ILC2s. And we did it in multiple ways. So the first one was by performing surgical ablation. We've done this in a way that we did cut the genital femoral nerve unilaterally, and then we analyzed the visceral adipose tissue of the site that was where we made the surgical ablation from the sham control. So the first thing we observed, and we got very excited with this, was that when you ablate the sympathetic innervation by surgery, we got a significant reduction of GDNF expression, and this beautifully correlated also with the downregulation of ILC2 activity in the gonadal adipose tissue. This was true by, or valid with the surgical approach, but also with the chemogenetic approach using DREADs. So if we now inject a virus that drives the expression of a DREAD receptor that has got an inhibitory capacity, so whenever we inject CNO, we block the firing of these sympathetic neurons, what we observe basically mimics or phenocopies what was observed with the surgery. There's an immediate reduction of GDNF in the gonadal adipose tissue, and as expected, this beautifully correlates with a reduction of innate type 2 cytokines. And if we do a chemogenetic activation, now with activatory DREADs, in a similar approach, so we inject these in one of the gonadal adipose tissue depots, now we observe the opposite phenotype. So activation of sympathetic neurons leads to increased expression of GDNF, and this beautifully correlates with increased ILC2 function in the gonadal adipose tissue. So clearly indicating that this aortico-renal adipose circuit can connect or connects to the brain and also controls ILC2. So activation of these neurons leads to increased GDNF expression and type 2 cytokine production, while inhibition of these neurons actually leads to a reduction on the firing of these neurons, reduced GDNF, and reduced ILC2 activity. So I hope I have convinced you, this is still unpublished data, that these data indicate for the existence in the visceral fat of a neuromesenchyme unit that actually in a concerted manner can control ILC2s, and in turn, this regulates obesity. And this all operates via a brain adipose circuit. In other words, there are cell bodies of central neurons sitting in this specific area of the hypothalamus, the PVH, that actually send signals to the aortic-renal ganglia that in turn projects to the gonadal adipose tissue. Here, the sympathetic neurons via the beta-2 receptor activate local mesenchymal stem cells to produce GDNF. That in turn is integrated by ILC2s that start producing large amounts of IL-5, IL-13, and metenkephalin peptides. I didn't have the time to show you this data, but the end result of this is that these cytokines create the appropriate environment for adipocytes actually to increase the expression of energy expenditure genes, including UCP1, amongst other, that leads actually to reduce fat mass and therefore increase resistance to obesity. So this is really my last slide. I hope I have convinced you by presenting both these published and unpublished data that neuroimmune circuits and neuroimmune interactions are very important, not only in barrier tissues or in barrier organs such as the intestine or the lung, where it can actually provide not only a protective immunity, but they can also contribute to the homeostasis of these organs. But we start seeing the clear examples of the importance of neuroimmune circuits to establish and preserve organismal physiology. And I gave you two examples of long-range neuroimmune circuits whereby the fine neuronal structures can sense environmental cues and then provide and translate those cues into distinct cell types, either directly to immune cells or to the coy cell types, such as mesenchymal stem cells, in this last example in obesity, that then control immune cell functions. So rather than looking at these two systems as separate identities, I like very much this painting from Leonardo da Vinci, the allegory of pleasure and displeasure. And I would like to argue and to propose that actually to maintain health in a complex beings such as mammals, these neuronal system and immune system are essential to preserve that identity in this neuroimmune that I would call the neuroimmune system in a very collaborative and productive manner. And finally, actually, this is my last slide, the place where we're based in Lisbon. Portugal is a beautiful setting, as you can see. I have to acknowledge the people involved in the work, Julie and Vanya, that are not in the lab anymore, but that push forward the project related with NMU and ILC2s. Cristina Godinho Silva that push forward circadian regulation of ILC3s, together with Rita Domingos, and finally Filipa that single-handed push forward the regulation of ILC2s in the visceral adipose tissue. Some of our collaborators to this work, and obviously the funding agencies. And I thank you so much for your attention, and I'll be more than happy to then reply to potential questions via Twitter.
Thank you, Henrique. Really, without words, your work is terrific, is incredible. I was really amazed by every step you took in the different projects. It's really incredible. Thank you so much for sharing the talks and especially the last unpublished data. Thank you for sharing. Henrique, as you we will guide people to ask questions via Twitter. So let me share my screen so we can go to this slide that we always post to ask questions for via Twitter to Henrique. Please go and search for the account Global IMMUNOTALK talks, and you will find a tweet that is already there that says, ask questions for Dr. Henrique Veiga-Fernandes here, and reply to that tweet with your question mentioning the hashtag Global Immuno, and Henrique will use the Immuno account to answer the questions. And remember, you can ask questions right after this live talk, or if you watch the talk in YouTube, you can ask questions in an asynchronous manner, and Henrique will reply contingent to his availability. But yeah, Henrique, again, thank you so much, and congratulations to you and your team for such amazing research.
Well, thank you very much. The pleasure was all mine. Thank you.
Okay, take care, everyone, and see you next week.
Bye. Thank you.
Bye.